morning everyone and welcome to today's Next Top Bioinformatics webinar. Amplicon Nanopore Sequencing Data Analysis using EPI2ME. Thank you all for joining us. I'm Ming and I'm the moderator for today. To make sure everyone has a great experience, here are a few things to know. This session will run for about 50 minutes and we will have a Q&A session towards the end. If you have any questions during the webinar, please feel free to submit them through the chat box, and we will address them after the presentations. Lastly, please keep your microphone muted throughout the session. In today's webinar, we are happy to welcome Ms. Jia Xun as our speaker. As the Bioinformatics Manager at NextGene Scientific, Jia Xun has spent over a decade in her field. Today, she is going to walk us through the Amplicon Nanopore Sequencing Data Analysis using the EPI2ME software. Without further ado, let's start the webinar today. Jia Xun, the floor is yours now.
Hello, good morning everyone. Let me try to see if I can access my slide here. I hope my sound and also the slides coming through to your side okay. So if everything's look okay, then we will start soon. So the slide today that we're going to talk about will be the Amplicon sequencing. So it's Amplicon sequencing. During these sessions, we will go through about more about the applications on Amplicon sequencing. But before we talk about the Amplicon sequencing, we will be looking into some backgrounds in terms of the nanopore sequencing. So if you have joined our sessions last week, I think you can skip for the kind of the first 10 minutes because we are going to recap or rehash the information that we have for the Oxford Nanopore Technologies and also the how the technology works, the devices, and etc. So before we continue to talk about the Amplicon sequencing and also the analysis of the Amplicon sequencing data. So the sessions you might be seeing is, if let's say there's any questions regarding the sessions, please feel free to drop your questions to the chat box. So nanopore sequencing. So let me introduce Oxford Nanopore Technologies. So this is a UK-based company, and they develop and also sell nanopore sequencing products, including the DNA sequencer, the MinION or the ProMe-ION, and also they have the solutions for sequencing DNA for different applications. So these Oxford Nanopore Technologies or the DNA sequencing technologies, basically they have been validated by more than 125 different countries. At the moment, or I think for the past 2 years, they have actually achieved more than 14,000 of publications where users are using technologies to study their kind of work for their research and also projects. So at the moment they have sorry, they have developed multiple sequencing devices. So including from very small scales all the way from the palm size sequencer to very high scale like printer machines shown pictures. So we will be going through some of the devices available by Oxford Nanopore. So the users of the technologies for this nanopore sequencing, they have used it to understand the biology or the developed methods that might be used for routine tests. So it has been applied in many different communities, and then it has been used in many different environments, including the space. So they have brought the device all the way to the space station and then have tried the sequencing there with microgravity. So it has been tested to work in many extreme different environments, and it also has been applied in many different industries for genetic testing or plant and animal genome sequencing, and etc. So this is a very flexible technology that can be used for many different domain of research. So the benefits or the outstanding features of the Oxford Nanopore sequencing basically it can provide more insights. So what we are seeing here, you can see actually we normally call the Oxford Nanopore sequencing as long read sequencing, but that is not quite accurate because nanopore sequencing can sequence all the way from very short DNA fragments, perhaps from a few tens of bases, like 50 bases or less than 100 bases. The DNA can be sequenced in Oxford Nanopore devices, and then all the way to very, very long or very, very large DNA molecules up to MB in size. So it's actually flexible in terms of the fragment size or in terms of the read length. So it can also sequence directly the DNA or RNA molecules. So the good points about this will be we are able to detect the methylated or the epigenetics variations inside our samples. So if there are any methylated C or any methylated A in the DNA molecules, then from the sequencing natively, we are able to detect the methylated bases. So from there, we can also directly get the DNA sequence together with the epigenetic information. In addition to that, so far I think it has also very fast turnaround time, and then it's very portable. We can carry it and then to do the sequencing in many different environments, and then it's kind of it has the real-time workflows. So this is also quite different from other sequencing technologies. So because the sequencing or the base calling happen real time, so we are able to get the data before even the sequencing process is finished. So it's like we put the samples into the sequencing machine, and then within 1 hour or more, we can start seeing the data coming out from the sequencing, and then we can start analyze the data. So this also helps us to observe the status of the sequencing. For some of the applications, we can kind of decide when to stop the sequencing based on the available data. And then this can help us to cut down the sequencing time and also the sequencing resources. Also, at the moment, I think the Oxford Nanopore sequencing solutions are one of the most affordable one, and then it can be kind of easy to get for most of the users. And then it comes with different scale, so depends on the requirements or depends on the demands. Users can choose whichever solutions that work best for them. So how the nanopore sequencing work? Uh, the the one that you are seeing in the slide here are the nanopores. So the nanopore sequencing, based on the name, I think you can also guess the sequencing basically happens in the protein that forms the pores, and then this protein is very small, so it's called nanopores. So these proteins are in nature, it function like a gateway between two systems. So this nanopore is fixed on a membrane, and then inside the membrane, kind of the membrane basically separate the outward samples, and then with another segment. So when the DNA molecules pass from one region or one sections to another through these nanopores across the membranes, the signals will be captured by the nanopore sequencing device. So based on the signals captures, the signals can be converted into bases. So from the signals, we try to see what are the bases that is passing through the channels, and then that is our basically our sequencing data. So this is the uh, animated versions of the nanopores. So from the left-hand side, the pictures basically show how the nanopore is positioned on the membrane. And then the DNA or RNA molecules is brought over to the nanopores and then unzipped by the motor enzymes. And then after unzip, the single-stranded DNA molecule will pass through the nanopores. And then when the process happens, as you can see on the right side, when the DNA molecule is passing through the channels or the pores at around 400 base pair per second, the electrical signals will be captured by the sequencing device, and then at the same time, it will also be converted into base calls. So the one that we normally see as ACTG during the sequencing. So the features of this is because it's actually direct sequencing on the DNA or RNA molecules. So it's kind of we can detect the signals like the methylated bases. So the C and also the methylated C might have slightly different electrical signals. So that's why we can directly get the basis information and also the methylation information directly from one sequencing run. So because this process also do not involve PCRs, so that's why the data that we obtain from nanopore sequencing will have no bias that come from the PCR or the amplification bias. And then all the sequencing and also analysis can happen at the same time. And then one of the very interesting features from this capability will be is named as adaptive sampling. So we will go to see what is adaptive sampling shortly. And then this one, basically all the chemistries or the pores, integrity, and everything has been tested. And then, so the sequencing process can be carried out by users quite easily. So the demand for the samples will also be quite straightforward. We do not really need the samples to be like in very, very, very good quality before we can do the sequencing. So just now we mentioned about the adaptive sampling. So adaptive sampling basically is a type of targeted sequencing. So these adaptive samplings only kind of at the moment apply in nanopore sequencing. So other sequencing technologies might not be able to do this because most of the sequencing technologies we only see the data after a sequencing is completed. But because the nanopore sequencing is a real-time sequencing, so while the DNA molecule is passing through the nanopore channels, we are able to tell what is the sequence of the DNA molecule that is currently being sequenced. So in the sequencing software, we can actually inform the software that what are our target regions or target genes. So when the sequencing is being carried out, the software or the sequencing software will check whether the one that is being sequenced are the targets that we want to get from the sequencing. So if let's say it's sequencing the right genes at the moment, then the whole sequencing process will continue. And then if let's say some of the pores are sequencing some non-target sequences or non-target molecules, then some signals will be sent to the nanopores so that the process or the DNA molecules that is passing through the pores will be ejected from the nanopores. And then the pores will be getting another DNA molecules to continue sequencing. So this is kind of a real-time targeted sequencing where we do not really need to perform any sample pre-processing like amplify the target regions or use the props to capture the target regions. So the sequencing device that are currently available from ONT. So basically all the nanopores are kind of put inside small items called the flow cells. So the one that you are seeing right now are two types of flow cells that come from ONT. So on the left hand side, that's MinION, and then the right hand side will be the ProMe-ion flow cells. So the difference between these two are actually their scale. So on the left hand side, the MinION flow cells is carrying around less than 2,000 of nanopores, and then the one in right hand side carry more nanopores. So the one on the left hand side, MinION is we are expecting it to generate around 10 GB to around 20 GB of data per sequencing run. And then for some applications like human genome sequencing, we need more than 10 GB of data or 20 GB of data. So in that case, we might go for the ProMe-ion flow cell, which can generate over 100 GB of data for one sequencing run. So MinION flow cell is suitable for small genomes or for applications that require lower data throughput. And then the primary ion flow cells is designed to generate large data for applications that require higher data throughput. So based on the selected MinION flow cells, there are multiple sequencing devices that can be used. So if we are using the MinION flow cells to do the sequencing, the options of the devices, there are two. So one is the MinION MK1D, the latest models that that we are having right now. This one can capture one flow cell at a time. And then there's also another device called the grid ion. So this grid ion also do the sequencing by using the mid-ion flow cells. And then at one sequencing run, it can gather up to 5 mid-ion flow cells. So it's like it can generate 50 to 100 GB of data per sequencing run. And then if we are having the primary ion flow cells, the sequencing device that we can choose, there are 3 at the moment. So primary ion 2 solo, primary ion 2 integrated, and primary ion 24. So from the numbers, I think you can also guess how many flow cell it can gather at one time. So primary ion 2 solo and integrated both can gather 2 flow cells at one sequencing run. So the difference between the solo and the integrated, as well as the device with uh, the blue icons that you see over here, those are the devices that comes with the GPU cards that can help real-time base calling of the data from electrical signals to bases. So for the device that do not come with the integrated GPUs, normally we need to have a computer that is installed with GPU cards, like perhaps the NVIDIA GPU card, or maybe a gaming computers that is equipped with a good graphic card. So yeah. And then if let's say we have a large number of samples to be sequenced at the same time, and then we need high throughput for each of the samples, then the options could be the primary ion 24. It can capture 24 primary ion flow cell sequencing run at the same time. So back to our topic today, Amplicon sequencing. So before I go deeper into how we prepare the samples or how the Amplicon sequencing workflows looks like, let me try to uh, explain a bit on the Amplicon sequencing for the context of genomics and in the context of metagenomics. So for our projects or our research, sometimes we might get the samples from environmental samples or perhaps microbiome samples from organisms. So this kind of samples normally is a mixed microbial community. So when we get the samples, we might have bacteria in the samples. We also might have virus and also fungus and etc. inside one samples. So these kind of samples, I think commonly maybe you might have soil samples, water samples, surface kind of the surface samples, and then stool samples, skin samples, and etc. So we do not expect to have only one types or one species in our samples. Yeah. So this is the common samples that we obtain for our projects or research sometimes. So when we want to study the DNA inside these samples, one of the way to do will be we can directly extract the DNA out from the samples by using the suitable extraction kits. After getting the DNAs, the total DNAs from the total samples extractions, we might go for amplicon sequencing, targeting perhaps one genes inside the samples. So all the, for example, like 16S genes, then all the organisms or all the microbes that are carrying a 16S genes, we are going to sequence the 16S genes out from them. The outcome from this amplicon sequencing are a set of data where it contains the 16S sequence come from many different types of bacteria. Another way of working on this DNA sequence will be shotgun metagenome sequencing. So this one, we don't care whether it comes from 16S or whether it comes from virus, it comes from fungus. So whatever DNA molecules that we gather from the samples, we prepare it into sequencing library and then sequence whatever that we can get from the samples. So this kind of sequencing or this application, the name will be normally named as metagenomic sequencing, and then sometimes people might be putting a shotgun metagenomic sequencing. So it's a sequencing of all the fragments or all the DNA inside the samples. So this is the DNA sequencing in the context of metagenomics. And then sometimes in our projects or in our experiments, we might be doing some isolations. So that means from one mixed samples, we could try to isolate different bacteria, different species into different cultures. So when we extract the DNA from each of these pure isolates, we are going to get multiple samples, and then each samples only have the DNA coming out from one single species. So when we perform the amplicon sequencing for these kind of samples, we are amplified the perhaps 16S genes from the samples. Then after the sequencing, we will be getting the data separately for each of the isolated samples or pure isolates. So this kind of application, the name is also amplicon sequencing, but it's in the context of genomics. And then at the same time, I think we can also perform the whole genome sequencing. So we do not amplify certain targets out from our samples. We directly put in whatever DNA molecules that we are able to extract from the pure isolate. This application, the name will be whole genome sequencing. So you can see basically from the same samples, and then based on different approach of preparing or processing the samples, there are at least 4 possible applications. And then out of these 4, 2 of them are having the name of amplicon sequencing. So sometimes people might be confused when we simply talk about the amplicon sequencing. Then sometimes we need to kind of understand further whether it's in the context of genomics or in the context of metagenomics. So metagenomics and also the amplicon-based metagenomics, targeted metagenomics, all that we have covered in the sessions in previous week. So this week we'll be focusing on the amplicon sequencing in the context of genomics. And the whole genome sequencing, the bacterial one or the microbial whole genome sequencing, we will be going through the application in more details next week. So back to this week. This amplicon sequencing, I just want to clarify about their differences. So these 2 amplicon sequencing looks like same. So perhaps 16S rRNA sequencing for both contexts. But the one in the metagenomics context, what we want to achieve will be the taxonomic profiling. So we want to understand what are the organisms that are present in one sample. And then normally the amplicon sequencing in the genomic context, we want to identify what is the species in our samples, because from the mixed samples after we do the isolations, of course, there are other methods to identify what are the bacteria, and molecular typing will be one of the methods. So sequencing out some marker genes like 16S will help us to identify the species. So the difference will comes in when we perform the sequencing for these 2 applications. So normal amplicon sequencing, if we have pure isolates, normally we do not really need to sequence a lot. So I think most common way of sequencing the amplicons for pure isolate, we might extract the genomic DNA, and then do the PCR to amplify our target markers. And then if the target genes or the marker sequence is quite kind of not too long, normally we can submit for perhaps Sanger sequencing to get the sequence out from the target. So, and then if let's say we choose to use high throughput sequencing like nanopore sequencing to do the amplicon sequencing, normally we do not really need a lot of the data throughput. Perhaps roughly 1,000 to 1,500 reads or sequences for samples will be sufficient to help us identify the species. And then on the other round, other things, for metagenomics, the sequencing throughput required would normally be much higher because one sample it contains multiple organisms inside, and then for each of the organisms we are going to sequence multiple sequences. So the normal throughputs that we want to achieve will be perhaps 100 reads or 100,000 sequences per samples. So that's why understanding whether the samples are in genomics context or metagenomics context will be important to help us decide how much data that we want to get from our sequencing run. So of course, different sequencing approach or different application, we will be requiring different analysis pipeline to help us work on the interpretation of the data. So for the genomic amplicon sequencing, uh, Oxford Nanopore do have developed a specialized pipeline for that. So the pipeline is named as WF Amplicon. So this pipeline is expecting us to input in data that come from amplicons, and then the organisms are expected to be haploids. So these particular pipelines do not intend to be used for deployed samples, and it's actually not suitable for marker gene sequencing or amplicon sequencing that comes from samples that is a mixture of communities. So if you have samples coming from environment, you extract the total DNA, and then the total DNA actually, the DNA is coming from different microbes, different bacteria, or different members inside the samples. These workflows will not be suitable. And then if let's say you have sample like mixed communities, another workflows will be more suitable if you perform the amplicon sequencing or amplicon metagenomic sequencing. So the other workflow is called the WF16S. This one has been covered in our session in last week. So yeah, I hope I managed to clear up some of the doubts that you might have if you are running some amplicon sequencing. So it's important to uh, design the experiment so that it's kind of we have a clear pictures whether it's in the genomic context or metagenomics context. The workflows that we are going to see today, basically, we are more focusing on the genomic context. So we are expecting the samples come from a pure isolate or come from a single organism or single species. We do not expect this particular workflow do not expect to have mixed samples or do not expect the sample to have kind of differences. So this you can consider this workflow as a successor to Sanger sequencing. So normally we have when we have an amplicon, in this case, we normally could be choosing Sanger sequencing to perform to help us identify the sequences of the DNA molecules. So why this Oxford Nanopore amplicon sequencing could be a good successor or even better in many cases. So it can actually be more robust compared to Sanger sequencing because Oxford Nanopore sequencing can sequence DNA molecules that are much longer. So the limitations of Sanger sequencing will be roughly, we will be able to normally get amplicon sequence that is around 1000 kb in size. But if you have amplicons that is longer than 1000 base pair, normally it will hit the limitation of Sanger sequencing. And then in that case, Oxford Nanopore sequencing will help us to resolve the amplicon sequence that is much longer than 1 kb. So Oxford Nanopore sequencing, it can sequence multiple kb of amplicons, and then it actually with the read length or the size of it, it can also help us to do the phasing or haplotyping, even though the our workflow do not expect to have deployed samples. So some of the benefits of Oxford Nanopore amplicon sequencing over Sanger amplicon sequencing are tabulated in the tables here. So, and I think if you are familiar with Sanger sequencing, you might have seen the limitation of Sanger sequencing in terms of sequencing amplicons that is of high GC. So, and then amplicons that is having some repeats will also cause some problems to the single sequencing. And then, of course, other information like if let's say there are some differences or some variants like the heteroplasmic variants or heterozygous variants inside the samples, then sometimes single sequencing might not be able to tell us exactly how the variance looks like in our samples. So this limitation is can be solved by using the nanopore sequencing. So let me show you slightly more about how the things looks like. So heterozygous variance detections, actually the single sequencing most of the times will still be able to help us determine if let's say there's any heterozygous variants in our samples. But if let's say there are heteroplasmic variants, so that means the allele frequency, or if we have like something like 60S amplicons, some of the variants, it might not be, it might be rare. So maybe 10% of the DNA molecules is having A, and then another 90% is having T. So in single sequencing, we might see a small peaks to show the minor variants, and then because the peaks might be very small, and then sometimes we might think it could be a background or it could be some noise. So for nanopore sequencing, because we are able to tell how many sequence is carrying the A, how many sequence is carrying the T, so from there actually it can help us resolve better in terms of the variance that is inside our DNA molecule samples. So normally if we have nanopore sequencing data, we can use a program called IGB or genome browser to help us to kind of summarize the data that we have from the sequencing. And then at the bottom parts, basically that is the typical single sequencing data, how it looks like. And then normally a good data, a good base that has been called in the that single sequencing data should have very sharp peaks. So for the one that we are seeing right now actually is a bit noisy. So sometimes it's a bit hard if you want to determine the sequence based on the single sequencing data such as this one. So after having these applications from Oxford Nanopore sequencing, what we can do by using these applications. So one of the kind of frequently applied scenario will be in the biopharmaceutical research and development. So sometimes the projects might involve engineering of cell lines and then gene therapies involving the CRISPR-based editing tools. And then with the flexible Amplicon sequencing from ONT, it can help us to do some validations like sequencing the guide RNA to check whether it's of our designs, and then characterize the CRISPR editing outcome, and then high throughput screening of antibodies, libraries, and also construct the verification in viral vectors and clonal stability checks. So this can be applied directly in the lab to help us kind of check the whole experiment. So how the amplicon sequencing is carried out, normally involving multiple steps. So starting from extractions of the DNA, total DNA from our samples, we can proceed to perform PCR to amplify our target genes or target amplicons. So this one, it could be kind of the recommendations is we design our primers to include extra bases upstream and downstream of our targets, because this one can help us capture the targets better. So in case some of the kind of the ends of the DNA molecules is not sequenced at a very high quality during the sequencing, then we still have some buffer bases to help with that. And then the protocol that has been tested by ONT, they have tested the protocol for amplicons that is having the size of 500 base pair all the way to 5 kb in them. And then after the DNA and also the amplicons are prepared, then we can go for library preparations. So the recommendation is using the rapid bar coding kits. And then because we do not really need very high data throughput for Amplicon sequencing, so the recommendations is we can always try to multiplex up to 96 samples per MinION flow cell. And then in terms of the sequencing, because data throughput is required is not that high, so we can always choose the low throughput flow cells, MinION flow cells to perform the sequencing. And then the base calling will be recommended to be of high accuracy. And then normally we just need to generate perhaps 1,500 reads per Amplicon libraries or per samples. And then in terms of the analysis, the EPI2ME software that is developed by ONT, it can help us analyze our Amplicon sequencing data that come directly out from the sequencing process. So before I go into analyzing the Amplicon sequencing data, so perhaps let me just quickly introduce you the software or the solutions available for data analysis that was developed by ONT. So the development of the software basically help us with the third steps of projects, so the analysis part. So from ONT, there are two main software. One is MinION. MinION is a device control software. So this MinION helps us to handle the sequencing process. So it's like when you want to start a sequencing, when we want to stop a sequencing, and then how's the sequencing performs at the moment. All the nanopores are whether the nanopores are healthy inside the sequencing device, and etc. So all these things can be controlled and monitored by using the software called MinION. So if we are running sequencing in our labs, this basically is the software that we need to install in our computer in order to control the sequencing process. And then another software is called the EPI2ME. EPI2ME is the data analysis software. So this particular two software, they comes with graphical user interface. So they are point and clicks. So it's quite user friendly for all users. So we do not really need to have prior knowledge in common lines in order to operate these two software. So other than that, nanopore sequencing, we have a lot of users using nanopore sequencing. So that's why the bioinformatics tools developed to handle or to analyze the nanopore data, nanopore sequencing data, there are quite a lot of them right now. So many developers have shared their programs through public repositories such as GitHub. So for users who would like to use those bioinformatics tools, they can get it quite easily from the public databases or public repositories. The one developed by ONT basically is fully supported by ONT, and then the bioinformatics tools developed by the community normally will also be supported by the community or the developers team. And what are the analysis that we can carry out by using the EPI2ME software? So actually it has included or comes with multiple analysis workflows or pipelines. So some pipelines are suitable to be used for analyzing the metagenomics data, and some will be more specialized to analyze the human genetics, like getting the genetic variants out from the human whole genome sequencing. And then, of course, there are also workflows that help us analyze the amplicons, help us analyze single cell transcriptome data, and then there are also workflows to help to check specifically on perhaps plasmids. So I think at the moment there are more than 15 different workflows available inside EPI2ME. So this EPI2ME software is a desktop application. That means we normally download it in our computer, install in our computer, and then the analysis is carried out in our computer. So it's not web-based. So most of the time we can do our analysis locally in our computer without requiring any data upload to any cloud or any servers through internet. This EPI2ME software is free to download for all users, and then it pre-packaged with more than 15 workflows or 15 pipelines. So the setup is quite easy. So it's like the normal the programs where we download the installer and then we go through the installation setup. And then once it's done, then we can directly open the program to use. This particular software is compatible with Windows. It also can be installed in Mac computer and also Linux. Just some head ups for the Mac OS users. The software have no problem to be installed in the Mac computer, but some of the workflows, their elements might not be able to run on Mac computer, especially if the Mac computer is having the latest Apple chips like the M3, M4, or even M1, M2. I think some of the programs you might encounter some problems to execute them. But for Windows and Linux, as long as they are using the normal Intel or AMD chips, basically it shouldn't be any problems when we want to run the analysis. So you can also see at the bottom here, there's a message talking about we must log in to access the cloud functionality. So what about because I have mentioned this software is installed in our computer. So how this cloud functionality come in? So this analysis that is carried out in EPI2ME, actually we have options to run it locally in our computer. But in some cases, when our computer is not strong enough to handle some of the analysis, we can also opt to submit the analysis and then run the analysis in the cloud. And then the cloud actually is provided by ONT free for all the ONT users. So the process all kind of simply covered within the EPI2ME software. So you do not need to install any additional programs, or you do not really need to know how to upload data to clouds or how to set up the instance in cloud and etc. So everything is kind of run directly inside the EPI2ME software as long as you have the EPI2ME software installed in your computer. So this is a more specific or more details on what are the workflows available in EPI2ME. So for human genetics, there are 3 different workflows available at the moment. WF trio for trio analysis, most of the time is useful for cases like rare disease, where we have the parents' data and also the proband data, and then we try to see what are the genetic variants that is inherited from the parents and then could be contribute or associated with the clinical observation that we are seeing in the proband. And then other than that, we also have human variations and also somatic variations to detect the genetic variants in whole genome sequencing or targeted sequencing in the normal conditions or germline variant discovery, or in the cancer samples where we try to identify the somatic variants. And then some basic tasks like alignment and basecalling, and workflows for metagenomics analysis, whether it's a shotgun metagenomics or targeted like 16S metagenomics. And then there are also workflows for plasmids or clone validation and transcriptome analysis. And then for targeted sequencing, we have the WF Amplicon, which are the one that we are going to focus more today. And then there are also workflows to help us analyze bacterial genomes or any specialized pathogens genomes as well. And also there are some workflows to help us with genome structures, which are normally useful for large genome assembly. So, and then at the bottom, you can see the CPU required. So basically all these workflows, they required more on CPU rather than GPU. So as long as we have a normal laptop or normal desktop that is carrying perhaps 8 GB of RAM, then it will be sufficient for us to work on the WF Amplicon data analysis. So the EPI2ME workflows and all the resources are kind of available in the GitHub also. So in case you still prefer to use the command line to execute the analysis, you can also get the workflows, the next workflows, etc., all from the GitHub repository. So I will be going through some of the steps that we can do to analyze our Amplicon sequencing data. So starting from, let me show you how the sequencing data looks like after the sequencing is done in the lab. So let me quickly open up the folders to show you here. So when we have installed the MinION software to help us handle the sequencing part, during the installations, a folder name data will be created in the C drive of the computer by default. And inside this data folder, there are some additional kind of data or files that are required for MinION to work on its sequencing handling. And then during the sequencing, it will create one folder for each of the sequencing run. So the name of the folders will be based on the sequencing setup. So when we set up the sequencing and then we name the run as perhaps demo amplicon, then a folder names demo amplicon will be created in the data folder, and then whatever sequencing data that is generated will be stored inside the run folder accordingly. So let me try to see if I have a full set of it. So if let's say I created a sequencing run with the name of test nanopore nanop, then a folder will be named as test nanop. And then inside this folder, we might need to go several levels down in order to see the files that is created from the sequencing runs. So from one sequencing run, normally we will have the sequencing reports in different formats. And then the one that is in the format of HTML, we can open it up in any web browsers in order to view the kind of the outcome or the summary from the sequencing runs. And then inside this folder, we will also see multiple folders. And then the one that can be used to do the analysis or the input for our analysis would normally be the FASTQ pass. And then inside this FASTQ pass, because one sequencing run, we can multiplex many samples using the barcodes provided in barcodes provided by ONT. So during the sequencing, we can set up the sequencing in such a way is the real-time basecalling and real-time demultiplexing can be happen. So after we get the data, all have been separated into samples or different barcodes accordingly. So we do not need to process the raw data in order to separate out or do the demultiplexing to get the data for each of the samples. So we will have one folder for each sample or each barcode. And then inside the barcode folder, we will have multiple FASTQ files. That's because the sequencing happened real time. So for this case, every 10 minutes is actually outputting one set of data for us to analyze. So normally, if we want to analyze all the data that are generated from a sequencing run, we will be using all the FASTQ files for the samples. So in case you are getting your data from public database or getting the data from your collaborator or some suppliers, you might be getting one FASTQ file per sample. So that is because they have concatenated or put together all the FASTQ files that is generated in cases like this. So this is how the data looks like. And then let me try to show you where we can get the EPI2ME software. So basically, to run the analysis, we need the data, we need the software, and the software can be obtained from labs, the EPI2ME or the EPI2ME Nanopore Tech.com. So this is where we can get the software. So when we come to this site and then scroll slightly down, there's a download button. And then when we click the download button, we will come to this page where the installer of different operating system will be shown over here. So depending on the operating system that you are having for your computer, we can download the installer accordingly and then set up the software accordingly. So for some of the detailed steps on how to do the installation, at the bottom here, there's one installation guide. When we go into this installation guide, this basically will kind of have the very detailed step-by-step of what could happen when we set up the EPI2ME software. So just follow through the process, allow the computer to install the EPI2ME, and yeah, I think everything is kind of will be settled accordingly. During the installation process, sometimes computer restart might be required, so I will always suggest to do the installations while there's nothing else running in the computer. So this is how we set up how we get the sequencing data and also how we get the software. Once the two things are in the computers that we have, we are ready for analysis. So let me try to get my EPI2ME program. So this is how the EPI2ME software looks like after we have installed it in our computer. Once we open the program, it will come to the dashboard. Or actually for first time installation, I think you might be reaching a page where it asks for sign in. So you can proceed to sign up an account. If let's say you have not actually have any ONT user account, you can sign up with one free through the interface and then sign in accordingly. And then it will come to the dashboard or the home page over here where it shows the summary of our computer status. And in case there's any new updates for the software, it will be also showing a notification at the dashboard over here. And then some latest news from ONT, like what have been done, what are the kind of some hot news recently will also be shown at the dashboard over here. And normally I will also kind of take a look at the settings of the EPI2ME software to see whether the working directory are available or set accordingly. And then come to the local tab to check whether the system's setup is completed with the ticks over here. If let's say it's not complete or kind of perhaps the system has go into the dormant or sleep mode, then I can come to the local tab over here and then open the setup to wake it up again or complete the setup if let's say the setup is not complete. And then over here, there's also settings for system resources where we can set how much computer resources that we want to allocate for EPI2ME to use for analysis. So this one, normally I won't set it to the max. So I will leave some resources for my computer to run some normal Windows processors and etc. So this is my current setup at the moment. So we have the home buttons where it will bring us back to the dashboard. And then after these two buttons, actually we have only two buttons left. So one is to be used for analysis. Another one is used to view the analysis results. So very straightforward. To run analysis, of course, we go to the launch button over here. Once we are at this workflow page, it will show the available workflows in EPI2ME. So based on our objective or our goals, we can select the one that we want to use for data analysis. So today we are going to look into the Amplicon workflow. So as you can see, the icons over here is light up. That means the software has been, the workflows has been installed accordingly. And then the cloud icon also light up. That means I have signed in my account and then I will be able to use the ONT cloud in case I want to run my analysis by using their cloud resource. So in some cases like human-related pipelines, it actually do not allow us to upload any human data to their cloud resource for privacy purpose. And then in some cases, perhaps the workflow has not been installed in our computer, then we will see the computer icon being grayed out. So if let's say the workflow set what you want to use is having a grayed out icon like this, we can actually proceed to click on it and then do the installation accordingly by clicking the install button over here. So once it's installed, supposedly the icon of the computer will be light up like this. So for the pipelines that has been installed in the computer, we will be seeing the documentations directly from EPI2ME, such as this case. So it will give us some introductions about these particular workflows. And of course, if you want to know more details, we can also take a look at the pipelines overview here to check what are the software is used in each of the step in the pipelines. And then on the right side, you can see at the moment by default, all the analysis will be carried out locally, run locally. And then if we want to use the cloud provided by ONT, we can always switch our tab to run in cloud. And then actually the interface will be almost the same. So how to set up the run and how to execute the run will be the same. So back to, because I'm going to run the analysis in my computer, so I switch it back to run locally. And over here, before I start the analysis, just want to show you that there are also other options to help us manage the pipelines that has been installed in our computer, including like running a demo analysis just to check whether the workflows has been installed accordingly, or perhaps we just want to check how's the pipelines looks like, whether it can be executed successfully by using the demo data. So we can also check for updates for the pipelines from here, and then switch different versions. If we want to kind of use different version of pipelines, we can also do the switching over here. And then in case we do not really need the workflow anymore, we can delete the workflows from here to clear out some storage space. And then if you want to check what are the files that are included in these pipelines, we can open up the folders to take a look at them. So at the moment, the versions that I'm having for the Amplicon will be version 1.2.2. And to run the analysis, I just need to click the launch button. And then I will come to this analysis setup page. So all the settings have been pre-configured. So most of the time, I just need to select what is my input file. So input files, we do have two options. So one is file, another one is folder. So the one that I'm having is going to be like a folder set. I have multiple folders because I have multiple samples. So for that case, maybe let me show you quickly about my data. So for this analysis, actually my input data, I have two samples, and then I have stored them in kind of each sample is having one folder named as barcode 01 and barcode 02, and then both these folders are stored in the folder named FASTQ. And then at the same time, I have also prepared a reference files. So basically, this Amplicon data it's actually a sequencing data for multiple genes. So the samples are actually mycobacteriums, and then the sequencing has amplified the kind of sequence the amplified target genes, including the uh, GYRB and then the ROB and also some other genes. All these genes are actually known to associate with multi-drug resistance. So in this experiment, I try to amplify the genes of the mycobacterium, and then I try to see whether any variants are found in the gene, and then whether they can help to explain uh, whether there's any multi-drug resistance in the mycobacterium for these particular samples. So this is how my demo data looks like. So back to the workflows, just to explain slightly more. Actually, this workflow contains two modes. So one is the variant calling mode, and then another one is de novo consensus mode. So if you would like to perform sequencing or Amplicon analysis similar to Sanger sequencing, we can choose the de novo consensus mode in order to get the consensus sequence out from the Amplicon sequencing data. And then in my case, I'm more interested to know whether the target regions or target genes in my samples is having some mutations or having some variants. I have actually chosen the variant calling mode. So yeah, basically this is the available options for us to analyze the Amplicon's sequencing data. So back to the EPI2ME program. Here I'm going to choose the folder as my input, And then inside this FASTQ, I have two samples, each samples one folder, and then I'm going to put in the whole FASTQ folder as my input folder. And the pipeline will help us analyze all the samples inside the folders. By default, even though we have, it could sequence a lot for our samples, but the pipelines will help us down samples to only analyze 1,500 sequences from our sequencing data. And then another options that I want to put in will be the reference file, because I want to do reference-based analysis. So I will select the reference in the FASTA format over here, so that it will proceed to perform the reference variant calling instead of give me a consensus sequence out from the data. So other options we can also take a look. So including like pre-processing. So it depends on what is the Amplicon size of our samples. So we can perhaps increase or reduce the read length according to our Amplicon size. And then of course we can also set the maximums, and whether we want to set certain cutoff in terms of the quality and etc. Other than that, variant calling options, we can set how much coverage that we want to set. We want to call the variants, and then for de novo consensus options, we also have other parameters that we can tweak in order to help us with the getting the consensus sequence out. So output options also depends on user preference, or else we can leave it as default. And then advanced analysis, similar, or we can also optimize accordingly if required. And then miscellaneous, I think that one depends on how much computer kind of additional settings on the computer resources. So by default, it's going to use 4 CPU threads. And then if let's say we have multiple samples, it will run multiple process at the same time. And that will be controlled by the next flow, and then based on the total allocated resources that we have in our settings. So not to worry that you have too much samples and then it could jam up your computer, because the next flow is the workflow or the task controller in our computer, so it will help us to manage the workload for the analysis run accordingly. So, and then the last part will be the next flow configuration. Here, I just want to show you that for each of the sequence analysis run, EPI2ME will try to assign a name to it, and then this name is actually random. So if you want to have a more meaningful name, we can always write the name based on our preference. So once everything is in green text, actually it's ready to be run. So we can proceed to launch the workflow accordingly. Yeah, so it will try to help us check whatever required data or files for the run, and then once it's in the running step, then it will come to the analysis page over here. So while waiting, let me show you the results page over here. So for all the analysis that have been carried out, it will be shown in the results page. So we can see the analysis run based on the name of the run, or and then the workflows that is carried out, and then the date of it. The status will tell us what are the analysis that are being carried out or that has been completed. So this analysis, Amplicon analysis won't take very long, so I will let it to run, and then we will come back to look at the results soon. So while the analysis is still carrying it out, so we can try to see the progress over here by clicking the analysis run. So you can see the task that is to be carried out in the pipelines is shown over here. So it will show the progress real time. So yeah, and then at the last tab over here is the logs. So this one, so in case some of the process run into problems, we will come to this particular logs tab to try to see what could be the reasons that cause the analysis to fail. So if let's say everything runs smoothly, normally we can ignore these logs directly. Okay, so the status now has been changed to completed. So we will switch back to take a quick look of the progress. So everything looks okay. And then the analysis time is roughly 1 minute plus. Once it's completed, we can try to look at the reports over here. So this is the Amplicon analysis report. So in this report, it will try to summarize whatever that we have for the run. So including like how many samples we have and then how many amplicons that we can detect based on the data or based on the information that we provide. So for this analysis run involving 2 samples and 5 amplicons. So for the 5 amplicons that we put in, actually only 2 were detected in the samples for barcode 01, and then actually another 3 amplicons are actually sequenced for another samples. So we can look at the summary of the samples here. And then of course there are some additional information like how much data that is analyzed in the run. So how much have been pre-processed on down samples. And over here it will show the sequencing data, sort of sequencing data QC. So how is our rate quality obtained from the sequencing run for sample 1 and also sample 2. And what is the rate length overall. So because we have multiple amplicons, so each of the amplicons are having slightly different sizes. So we are seeing different peaks because different amplicons are being sequenced for the samples. And then the how much data that we have for the samples. And then the summary will be like for each of the samples, how many rates are kind of analyzed and then detecting how many amplicons. Of course, we can also see the summary by amplicons. So EPI2ME genes, how many rates have been discovered to be of these particular targets, and then how many of the CAT-G and etc. And then the details, the more details one will be, the breakdowns will be over here. So to see barcode 01 is having the RPOB sequences, and then how many reads are belonging to these particular amplicons, and then what is the median read length and etc. And the coverage for each amplicons over here. And importantly, this is the objective for my analysis. I want to check for each samples and each applicant, what are the variants that I can find in the genes or in the targets. So like the CAT-G genes in barcode 01, we are seeing variants that change from C to G. And then this one I can further analyze whether it's associated with certain drug resistant phenotypes of the samples and etc. So at the end of the reports, we can see the programs that is used for the analysis, including their algorithms, and then the parameters are also shown here. And then the output files, because this is only the summary reports, all the output files, we can get them from the files tab over here. So the explanation for each of these output files is also available in the documentations that we see in the workflow page. And then because Amplicon sequencing data analysis involving sequence alignments. So there's a light version of IGV over here to help us quickly check the alignments of the data against the reference. So like this one is currently we are looking at the GIRB genes. For barcode 02, we are seeing a very good coverage or very good alignments of the data against this target. And then we are seeing variants being called, and then those variants are located in those locations over here. Over here, we can also switch to look at different amplicons, and then yeah, it's actually a light version. So if you want to have the full function of IGV, we can download a copy or download the software and install in our computer, and then view the alignments generated from these workflows accordingly. And then this is how we can get our analysis result files. And optionally, we can also click over here to check some of the information of the analysis run, including like renaming the analysis, or in case the analysis is no longer required, we can delete the analysis over here. And sometimes I would prefer to directly open up the folders of the analysis run to get all the files listed directly in my file explorer over here. And then the reports that we've seen just now uh, is also available as one of the output files. So in case you would like to share the results with your collaborators or your lab mates, you can directly share the HTML reports to them. And as long as they have web browsers application in their computer, they will be able to open up the reports and look at the summary results. So they do not need to install the EPI2ME program in order to view the reports. They just need to open it in any web browser. Okay, so I think that is all that I would like to demonstrate to you on how to analyze the Amplicon sequencing data, on how to handle the Amplicon sequencing data. So let me just quickly summarize what we have over here. So this session we have gone through on knowing what nanopore sequencing is about, and then the company of Oxford Nanopore Technologies, ONT. And then we have also looked into the EPI2ME program and also the WF Amplicon analysis. Before that, we also have tried to identify or kind of differentiate the Amplicon sequencing in the genomics and also metagenomics context. So with that, basically, I will be ending my presentation over here. So and then let us take a look at the Q&A.
Thank you, Jiaxun, for the insightful presentation on how we can use the EPI2ME analysis to analyze the Amplicon nanopore sequencing data. So now we'll move into the Q&A session, and our speaker, Jiaxun, will address to your question. You can unmute yourself to ask the question or type in the chat box. So now let's see in the chat box, there are several questions. I believe our field application manager, Chi Hong, has already addressed some of them. And another question from Joyce is EPI2ME has KEGG annotation for WGS.
So back to the questions regarding the KEGG. The one in EPI2ME for whole genome sequencing, it only have the structural annotation or genome annotation identifying where are the genes located in the genome, but it do not really have very detailed functional annotation, especially the KEGG. So I think in the next sessions we do have more details on the bacterial genome pipelines in EPI2ME. So welcome to come back perhaps in the next sessions, then we can go through in more details of what are covered within the bacterial genome pipelines. So quick answers would be sorry, no KEGG annotations at the moment.
Oh, right. So the next question is from Citi. Since both nanopore 16S metagenomics and Amplicon sequencing can identify the species, so what are the further difference between them?
Let me try to get back to questions again. So Amplicon sequencing identified the species and 16S also identified the species, right? So that one would depend on our samples. So if our samples is a mix of different bacteria inside, and when we sequence the 16S, then we will know the identity of the species together with the relative abundance inside the samples. So this one is in the metagenomics context. And then in the genomics context, then supposedly in our total DNA samples, we only have one bacteria. And then when we amplify the 16S, yes, we can identify try to identify the species of it. And the relative abundance information is not available, not because the sequencing throughput is not enough, but because the samples is supposedly to contain only one species. I hope that can answer the questions. And actually also want to emphasize one more thing. Even though the 16S has been used widely to identify the species in our samples, but because the identification is only based on one single gene that is around 1.5 kb in size. So for some species, when they are sharing or they are having very high similar 16S, we can maybe identify them up to the genus level, but it's a bit hard to tell in very details of whether they are belonging to a specific species. One of the example is like the Bacillus cereus. So under this bacillus genus, there are several bacteria or several species very, very close in terms of their evolutionary relationship or molecular typings. So when we sequence the 16S out of several very closely related bacillus, we can identify them as bacillus most of the time, but not able to tell specifically like whether they are bacillus cereus or bacillus anthracis or bacillus thuringiensis and etc. So there are also limitation in terms of using 16S to identify the species up to the very detailed species level. So I hope that can answer your question.
Okay, the next question from Sarah. For adaptive sampling on human samples, which workflows in EPI2ME should be used?
Adaptive sampling, is it?
Yes, adaptive sampling.
For adaptive sampling, supposedly we
On
should be using.
is adaptive sampling on human samples lah.
I'm trying to get the questions from the chat box. I think it's Amplicon sequencing, is it?
No, sorry. You mentioned in the earlier presentation, you mentioned about adaptive sampling as well, right?
Oh, yeah.
Amplicon sequencing and there's adaptive sampling. If I want to do adaptive sampling on human sample, I would just want to do adaptive sampling on some gene clusters. Which workflow in EPI2ME should I use?
Supposedly for adaptive sampling, we should be using the human variation workflows.
Okay, thank you.
Yeah. Actually, if let's say you just want to kind of use the adaptive sampling to target certain genes inside human genome, I think the human variation pipeline should be more suitable. So we need to get the human genome as the reference and then map the adaptive sampling data against the human genome and then try to get the variants out from the adaptive sampling data.
Okay, thank you so much.
All right, is there any more question from the floors? Yep, I think there is no more question and we can conclude the session. So thank you again. Sorry.
Sorry, I think that I saw another questions in the chat box. Using EPI2ME from the results of analysis, can I have abundance table, something like OTU, ASV, and taxonomy table? How as I need this for another analysis? So this is actually a question I think should be back to the sessions that we have last week. But I can have some quick answer on this. So we do have the abundance tables from the EPI2ME metagenomics or 16S analysis workflows, but those analysis, the abundance tables won't be in the OTU or ASV format or data type. So if you would need the ASV or OTU abundance table like biome files or other further analysis, I think we will need additional pipelines or programs to help us with that. Perhaps the CHIME tool will be able to help us cluster the sequence, the 16S sequence into the ASV or OTU first before we can work on the abundance table in the downstream analysis. So short answer to the questions, EPI2ME analysis, it won't be giving OTU or ASV. It only has the abundance table based on the species identified. I think that's the question. I think
Yeah, about the last question, can we have the recording? Yes, we will share the recording with the registrants and participants.
Minty,
Yes?
I think I would take like 1-2 minutes to because just now there was a question about what's the difference between nanopore sequencer and Illumina.
Oh, yes, you can further elaborate or explain to our
Okay,
participants.
so do you see my screen?
Yes, we can see your screen.
Okay, so I'm just doing a simple comparison that I have over here. So basically, just now I mentioned that for the two platform, so for the for the nanopore, my example is actually the MK1D MinION. Where else for the MySeq, because now they launched a new sequencer from Illumina, it's actually called MySeq I100. Okay, so just now I mentioned that they are different in terms of sequencing principle, the sequencing length, the output, the equipment size, then even let's say for application wise for 16S, their capability are different, then protocol, and then some taxonomy analysis, and then so on and so forth. Okay, so for the sequencing principle, because Illumina is using like the fluorescent base and then capturing, so therefore the method they call is actually called sequencing by synthesis using fluorescent labeled nucleosides. Whereas for nanopore, just now I think Jashin already explained. Basically, we are checking, measuring the electricity current as the DNA pass through the nanopore. And then also, as Jashin said, the read length are actually different between these two platforms because nanopore we can sequence DNA fragments from a size as short as 20 up to even million bases. Whereas Illumina system currently they have a fixed sequencing length, so ranging between 50 bases to up to 500 bases. Okay, and then depending on the flow cell being used, the output for MinION is ranging between 8 to 14. We have only one type of flow cell for MinION, but sometimes in an ideal situation, the output will be more, and in some of the not so ideal run, then the output will be slightly less. Whereas for MySeq, the estimated output is actually around 25 to 30, again depending on the the model of the sequencer as well as the flow cell type. Okay, so the size, okay, so MinION is very small because we need an external computer. So for MySeq, it's bigger. Then for the QC requirement, okay, and then the one of the main highlight is actually for 16S, because we can actually sequence fragments 20 up to 1 million bases. So therefore, we can no problem sequencing the entire 16S region of the bacteria. Well, for MySeq, their current protocol is actually only targeting the V3-4 region, which is actually approximately 450 bases. Okay, so yeah, I think I will stop over here. Okay, thank you, Minty.
Yeah, thank you, Chi Hong, for the explanation. So I will conclude this session. And yeah, your opinion matters. So please fill out the survey form as I paste in the chat box, and we will send the e-certificate to you. So thank you to all of you again for attending this session.
Maybe I think there's one more question. Yes,
sorry.
nanopore can be used for ITS fungus. Basically, we can sequence any amplicons, so including but not limited to 16S, ITS, 18S, even if you have a COI region, as long as anything that you can PCR amplify, we can actually sequence it under the protocol that I shared earlier, which is actually the amplicons. So therefore, we are customer sequencing like HLA, yeah, and also SSR, simple tandem repeat, that kind of things. Yeah, thank you.
Yeah, is there any more question from the participants? If you have any further questions after this webinar, so feel free to contact our product specialist, or you can contact us at sales@nextgene.my. So we will address them accordingly. Yeah, lastly, so thank you all again, and we hope we can see you guys again in our next webinar series. Thank you.
Thank you so much.